trying to cover too many varieties too quickly, so we're going to cut down the number of varieties for identification from now on. I think it's important to take down the notes that apply to these particular varieties. Apparently you're not either underlining them or paying too much attention to the important distinguishing features. Yesterday's class had quite a bit of difficulty. Very few students really got a good score. The only blessing was that everybody did pretty bad. I mean for you. So we're probably going to cut down to about 10 varieties of the whites and 10 of the varieties of the blacks, just using the most important ones. Now, at the last lecture, we were taking up rather rapidly some of the descriptive terms that we're using in variety description. And I believe we got over to the leaf. We have already used a lot of these characteristics to identify the leaf. Notice where I draw a leaf, it's in a pattern of 5. If you first draw the main veins, you can always draw a pretty good grape leaf. The central midrib is the longest, and the 2 side ribs and the basal portion of the rib structure. And then all you have to do really is to attempt to connect up these more or less, and you come out with what looks somewhat like a grape leaf. I should probably come over here a little bit of ways, make it look a little better. Now, if we take the leaf in general, these indentations here on the top are usually called the upper or superior, whichever word we want to use, sinuses. The sinuses refer simply to these openings. In other words, if you had an entire leaf, there would be no sinuses. An entire leaf would more or less follow this outline here, in which case you wouldn't have any sinuses at all. That's a very rare situation in Vitis species where you have an entire leaf with no sinuses. And then we have the other indentation at the base here, which is usually shallower than the upper sinuses, so it's obvious that you'd call those the lower, since they're on the lower portion of the leaf, or inferior sinuses. And then lastly, you have the large opening at the petiola junction with the leaf, which is right here, and this represents the petiola sinus. Now, as we noticed in the field, these can take on various shapes and types. First of all, this indentation may be very shallow. Shallowness means the distance from the outer periphery or the marginal line that you'd put around the leaf. If the indentation only goes in a short ways, we'd say it's shallow. When in a long ways, we would say it's deep. And then the width of the sinus is very important, because some sinuses tend to be more or less narrow and deep. Others tend to be flattened out. As you remember, the sinuses of the Zinfandel, for example, are very wide at the base. And sometimes the lobes may even come back so that the sinus is closed. In other words, you can see that if this lobe of the leaf were to come out thusly and this other lobe were to come over like this, then this sinus would be obviously closed. The same phenomenon of the closing of the sinus can occur with the petiola sinus or the inferior sinuses as well. Now, many of you, I think, in trying to identify a variety will assume that if you take one leaf, that this is going to satisfy the sampling and that this will give you a clear picture of what the variety is like. This is where we make a lot of errors because it's an average really that we're looking at, so it's very dangerous to take one leaf and try and classify it. In our work on description of varieties, we take a sample of 50 leaves to measure to get an average. So that's point number one. Just because you don't see a character on one leaf, you cannot give up at that point and say that this does not conform to the usual description. You've got to take a sample and look around. Now, despite all these fine features of identification in the leaf, most people who know varieties simply recognize them like they do people. You walk up to somebody and you recognize them, not because their ear is longer or their nose is shorter, but because overall they give you a certain impression. The vine is different in one respect. It never talks back to you. So all you have to really center on is the overall impression that you get from that vine, the leaves, the growth habit, the fruit, and so on. And once you know that, they get familiar. And once you've reached that plateau, you're going to know your varieties pretty well. Now, that hasn't arrived yet. We don't expect that you will reach that position maybe for some weeks or even long after you get out of this class. But at least you will have the basic framework with which to recognize varieties. That's the important thing. Of course, if you continue to get 0 grades in the quizzes, that wouldn't be too good either. Now, the second thing that's of importance is the hairiness. And you notice the hairiness of the leaf, or as we say, tomentum, is usually on the basal or the backside of the leaf. So you usually look at the backside rather than the face. The face of the leaf is usually quite smooth or glabrous. The hairiness then. As you've noticed in the field, there are several distinct types. One is a short tomentum that consists of a lot of short hairs that sort of are like whiskers on the leaf. So you get a uniform appearance of hairiness that more or less makes the leaf felted. And this is very characteristic again of the Zinfandel. Is a good example of this felted or short, stiff hairiness on the leaf. This also provides a lovely home for insects because the dust you see, since the hairs are more or less uniform, the dust and other detritus accumulates on the top and it makes sort of a little house, so the red spiders have nice little apartments down here below, and they're well protected from any spray or other difficulties. So the hairiness is important, especially in the resistance to various diseases and insects. Now, the second type of hairiness we might call, there are various names used, but I think the ones that are most descriptive, the easiest to remember, cottony type of hairiness. And you've seen this where the hairs congregate in sort of little tufts that look like bunches of cotton, little tufts of cotton. And this is quite typical of quite a few varieties, the cottony type of hairiness, where the hairiness is not really accentuated, but the few hairs that are present congregate into these little tufts, a cottony type of hairiness. And this means, of course, that the leaf is not nearly as hairy as the tomentum type because you don't have as many hairs to begin with on the surface. And then a third type we might call the arachnoid or the spiderweb type. Which again is a pretty common type of hairiness on these vines. And that is a long type of hair which is more or less twisted. So the appearance as you look at the leaf is one of these long loose hairs forming more or less a spider web over the surface. Now, it is true that in some cases these hairs may collect together and you may go over into this condition. It might be confusing in some respects, but in the early stages this type is quite different from this or from this. So in general, there are three types of hairiness, and then of course you have lastly leaves that have practically no hairs on them, for example, like the petite syrah, which we call glabrous. Now that again isn't strict. If you take it under a low power magnification, you'll find there's hairs practically everywhere on the vine. So you're using, again, an average situation that you probably only can recognize by gaining experience. Now the color is sometimes important. The color of the leaf petiole may be reddish or purplish. Sometimes this color extends up into these veins quite a way. White Riesling, for example, is rather peculiar that way because it is a white-colored variety, but the leaf petioles are very purple or reddish, which is rather unique for a white grape. So this is a good distinguishing character of the foliage of the White Riesling.
Is there a good way to tell whether it's just, I notice a lot of petioles are red, just looks like from sunburn.
Yes, the sun is usually a factor. In other words, if you're looking for this color, you've obviously got to go to a well-exposed leaf. And remember we said that in sampling. If you're going to get a typical leaf, it should be about the 6th to the 8th position on the shoot. It should be well exposed. You can't go in under the vine in the shade and expect to find a typical leaf of the variety. Now, I am not going to take time to go over the characteristics of the cane in detail. You have seen some of them, especially, for example, the difference in the buds and the difference in the color of the cane. An additional factor is that some canes at maturity are ridged. In other words, they have bands of tissue that are more or less ridged on them. This is of more interest in the identification of species than it is of varieties within, say, Vitis vinifera. So we'll come back to this at a later time when we begin to study species in more detail. Now we're going to get into a review of classification so that we may properly place the species in some order that we can readily study and understand. First of all, the large family of grapevines we usually refer to as the Vitaceae. Vitaceae. Other names have been used, but the designation given by Lindley was the one who proposed this name for the family in 1860, I believe, 1830 rather, 1830. This name has been retained now by most botanists, and so we can forget, I think, the other names that were proposed earlier or later. Vitaceae, which simply refers to the whole family of grapevines. In other words, the grape family. Now this family belongs to the class of plants called Dicotyledons. In other words, the plants that when they germinate have two seed leaves. As you know, this is a seed leaf, this is a seed leaf, two cotyledons. So it belongs to that general class of plants. When a grape seed germinates, you do get these two small seed leaves appearing above ground, the first leaves to appear. Now the order, it belongs to the large order called Rhamnales. And this includes several other families. I don't particularly care that you remember all the detail on these things, but in general, I think you should know in what groups we find the grapevines. The order then is Rhamnales. So our first classification would be the Dicotyledons. The class is Dicots. It's a review for your elementary botany here. The order is Rhamnales. And then we have the family Vitaceae, along with Rhamnaceae, Liaceae, and several others. Liaceae is related quite closely to grape, but they are mostly tropical trees. Although the flower looks somewhat like a grape flower, they're mostly tropical trees, and some authors leave this out. We tend to leave it out also, out of the Vitaceae. Now, these Rhamnales have within them some interesting plants. In the Rhamnaceae, for example, we've got the group of mountain lilacs, these shrubs that are very common in California, called ceanothus. If any of you are interested in ornamentals, these blue flowering shrubs that you see on the coastal hillsides and also in the Sierra, ceanothus, or mountain lilac as it's called, has this relationship to grape in that it's in the same order, but in a different family, in the Rhamnaceae. And there are many other plants that fall in this same group. Now, if we get to the family as a whole, there are certain significant characteristics that you would look for that might help you to identify whether the vine or plant is in the Vitaceae. And one of the most significant characteristics of the groups are the specialized shoots that we call tendrils, and these are borne opposite the leaves. These enable the plant to climb, attach and climb upward on other shrubs and trees. The botanist calls these climbing plants lianas, or climbers, L-I-A-N-A-S, which a word that you'll probably see, lianas or climbers, to distinguish them from other plants that do not have these climbing tendencies. Now, this would mean then that the Vitaceae are really related to a forest-type ecology. In other words, the vine has got to climb up on other shrubs or trees and makes a canopy up above them ordinarily, where they reach the light and sunshine. This also means that they usually thrive under conditions of high temperature and high moisture, because most of the forest trees have to be in areas where they have moisture the year round and have a considerable amount of temperature to grow well. Now, this leafy portion of the vine forms a canopy above. When it gets on, say, a poplar tree, the vine will climb up and eventually make a canopy up or over part of the tree. So actually then the vine is sort of suspended because the long trunk reaching the ground is fairly naked. There's not many branches here. It's shaded out. So you have these long, narrow trunks that are covered with a loose bark that are really the only lifeline between the nutrients and water in the soil and the active leaf surface above. They are also very tensile. They can be bent quite easily. Obviously, because if the tree were to blow in the wind or something, it would pull the vine right out of the ground. But if you see them, under natural conditions, a vine always has a sway or an undulation in it to take care of any of these eventualities. So this is rather an important point, that actually the vine is a forest plant associated with trees, with other shrubs. It does not ordinarily grow by itself. Some recent work in Australia is rather interesting in this regard, because it has shown that the vine, like some of our forest trees, has to have certain special organisms that grow on the root in order for it to thrive. These are Mycorrhizae. And this was demonstrated by simply growing small grape plants that were grown under sterile conditions in a soil taken from the forest that was sterilized. When this was done, the vine refused to grow very much, made very weak growth. However, if the soil from the forest were then inoculated into the sterile soil, the vine grew very well. They were also able to demonstrate that in the roots of the vine, we have these so-called organisms growing that aid the vine in its nutrition and in its growth. Is there a question?
Yeah. What would happen in, like, if you're fumigating a field or something, or is that going to kill off all these things? You're going to have less growth?
I couldn't answer that question, but I presume that a lot of the mycorrhizae would be killed off. Whether you would kill off enough of it or not, I don't know. But we have had this experience in the greenhouse many times in growing grape seeds, that if we use a highly sterilized soil and do not add another unsterilized portion to it, or some outside soil, we do get very poor growth. Even if you take peat moss, for example, and add that to it, that seems to be enough. So this is just one indication of why it's interesting to know what the natural antecedents of the growth of these vines are. In other words, why do these relationships come about? Now, similar relationships occur in pines, for example, in pine trees. They also are dependent upon these root organisms or saprophytes to assist in their nutrition. So then a vine has something in the way of a forest ecology, certainly, that would warrant some more investigation. Now, we mentioned the tendrils as being rather unique to the Vitaceae. Another interesting thing is that the flowers of this grape family are usually very inconspicuous. They're small, and they are usually characterized by having a corolla instead of being brightly colored as it is in many flowers. The corolla is greenish, usually 4 to 5 petals. But these petals, instead of opening outward, are usually connected together to form sort of a cap or calyptra. And in some vitis, these petals, instead of opening from the top, like you would expect, they open from the base of the flower. We draw the ovary in here. This cap covers the flower over, and the corolla, or the petals, are joined together to form this calyptra. C-A-L-Y-P-T-R-A. Essentially, it is a joined corolla, where the petals, instead of being separate, like they are in a rose or a daisy, the petals are joined together to make a cap that fits over the top of the flower. So when the flower blossoms, in many vitis species, the petals break clear at the base of the flower and open up, so that sort of an umbrella condition is formed over the top of the flower, which falls off as a unit. In other words, the petals fall off, still held together at their apex or their top. The seeds in the Vitaceae are bony in structure, with certain convolutions usually. You have probably looked at seeds in the field, but not under magnification. In the one genus Vitis, the seed has a shape, more or less a pear-shaped condition, but on the back side, this chalaza is on the ventral surface. On the back side now, if we turn it over on the back side, we get another condition here. We get 2 long furrows. These 2 long furrows are indentations of the seed, So actually, if we draw the seed in some detail, we will see more or less indentations going down into the body of the seed before, I'll get that right yet. These folds go well into the seed. And this folding of the seed is characteristic. In some cases, of course, it's much more complicated. Some of these genera in cross-section have folds practically all the way through them. And this, in some cases, distinguishes the difference between Vitis and the other genera. Yes?
If the corolla is small and usually green and inconspicuous, what draws the pollinating insects to it?
Well, the answer to that is the pollinating insects are not drawn to the flower. And for most cultivated varieties, at least, bees do not work the flowers except on rare occasions, so the flower has to be self-pollinated or wind-pollinated. So if you do have, for example, a variety that does not have pollen, a female variety, if you interplant it, you may end up in failure unless the wind helps you out. Sometimes the wind is effective, but in the northern European countries, for example, where there is little wind at the time of blossom, these female varieties are practically sterile. They get no fruit at all. Now the fruit is described as a berry. It's soft and a juicy fruit. The embryo is usually just a little spindle-shaped structure here that is not well developed. It's found right at the very tip of the seed and is a little over 1 millimeter long, so it's very difficult to see or to observe because it's practically surrounded by this bony or flinty-like seed coat. It's very difficult to try and dissect out the embryo of a grape seed. The embryo remains small and spindle-shaped until the seed actually begins to germinate or to grow, and then the embryo develops and absorbs the oily or fat-like endosperm. The endosperm, the white tissue in the seed forming the bulk of the seed, is a fat storage organ or an oily endosperm, so-called, which again is characteristic of this family. Now, the family is a very large one. It comprises over 1,000 species that belong to this grape family. The geographic distribution of most of these is largely tropical or semi-tropical. And there are no living species in the Arctic or Antarctic regions. In other words, the temperate zone is the limit of the distribution of species. It is interesting that practically all of the important cultivated species of the family come from the temperate zone and have a deciduous type habit. In other words, the leaves fall during the cool season. A complete enumeration of the species is far from realization. Their distribution is over 80% Asiatic, African, and Indonesian. In other words, in the oriental tropical areas and in Africa, most of these species of the family are found. Fossil records indicate, especially from seed remains, that the family was present in the lower Eocene. Now, I'm not going to review paleontology for you, but I expect you to look up what the lower Eocene epoch is and see how many millions of years ago that is. That's an assignment for you now. The lower Eocene. So one thing is plain from this, that this grape family has been around for a long time. The past distribution extended much further north, because fossil remains have been found up to 70 degrees north of the equator, which puts them really into today's Arctic region. These fossil finds of seeds have likeness with 4 genera that we know today. That is Vitis, Ampelopsis, Caratia, and Tetrastigma. There are 2 extinct grape genera that have been described to the paleontologist, which don't correspond to anything we know today. One's named Paleovitis and the other is named Cystites. They are grape-like, but they cannot be equated with any of these genera that we have listed. There have been many other fossil finds, but some of the interpretations were in error because when they were based on leaf prints, further examination by more recent workers has shown that some of the fossil experts got mixed up with sycamore leaves and other similar prints which they had erroneously placed into the Vitis family, into the Vitaceae. So from this evidence we can at least say that the family is a very ancient one of the most ancient of these woody genera of lianas, of climbers. Now the partition of this large family began first with Linnaeus in 1753, and I think it would be easier now to follow what we have on the board and take the division of the grape family step by step. I have placed it here chronologically because I think it may be of some interest to follow it in that manner, although most of the listings are not done in this way. Vitis, you can see that the common genera known earliest would obviously be the first ones described, and that's what you find, that Vitis was described by Linnaeus and published on in 1753. This was the first general compendium on plants, as you know from your botany. About 60 species have been described to date that are in the genus Vitis. The next group is Cissus, which was also described by Linnaeus, and this is a very large genus, 320 species. And we must say here that many of these tropical forms are not yet fully known or described. There are probably many more to come because there has been very little work done in the tropics. And then we come to Ampelopsis, which Michaud described in 1803, 23 species, Caratia in 1818, 61, and so on. Now, we might notice here that there are 2 genera here in which only 1 species has been described, so they're relatively unimportant unless they have some further interest from the evolutionary standpoint. So you really have 4 genera that are rather unimportant, so that reduces us down to about 10 genera that perhaps are of more interest. Now, the relationships between these different genera are often complicated, not clearly defined. We have, I might say here, the last type to be set aside was done by Alston in 1931, and he pulled this out of the Cissus group and set up a new genus with 230 species in it. One of the last was done by an American, Small, who thought that the muscadinia, or the so-called muscadine grapes of the southeast, are so different from Vitis that they should be in a separate group. And I think we would like to go along with that idea. So this becomes a new genus, Muscadinia, and we have 3 species in that, which we'll detail in a few moments. Now this Muscadinia group is different in several respects. It has, for example, three species, and these are the rotundifolia, which you've already seen in the vineyard. There's a good collection of rotundifolia varieties out there. Munsoniana, which is sort of a small issue of the rotundifolia, smaller leaved and smaller fruited. And then there's one from northeastern Mexico, Hopinoi, which we do not have in the collection. We had it at one time but lost it. So we only have 3 species. Now these have 40 chromosomes, whereas all of the Vitis species have 38 chromosomes. So on the basis of the chromosome number, then, we could easily separate these 2 groups as well. Now, certain of these groups or genera, early defined on morphological grounds, such as the number of petals, the structure of the seed, and so on, correspond reasonably well with chromosome number, and perhaps further work will show that chromosome number in itself may be a very good means of segregating or differentiating between these various generic groups. For example, Muscadinia, Parthenocissus, Ampelopsis, and Ampelocissus are fairly closely related. Ampelopsis, Ampelocissus, Parthenocissus, and Muscadinia have many characteristics in common, and it is interesting that all of these have 40 chromosomes. All of these have 40 chromosomes. The genus Cissus, the second one on the list here, is a very large group. It has many species with a chromosome number of 24 and some with a chromosome number of 26. This is a very large assemblage of many different things and will undoubtedly be further segregated into a number of other genera. And then we come to Cyphostemma, which you can see is a very large group. And these are interesting from the evolutionary standpoint because they contain species with the lowest number of chromosomes. 2n equals 20 or 22. We mark these over with the X as having 40 chromosomes, the Vitis 38. Cissus is 24 to 26. And then finally the tetrastigma is characterized by still another chromosome grouping, although there are certain exceptions to the rule, but tetrastigma has 2n equals 22, and sometimes there are polyploids or double numbers going up to 44, but the lowest group there is 22. This would indicate that if we take chromosome number itself as a measure of evolutionary development, the lower chromosome numbers being more primitive, then we would assume that the Cyphostemma, since it contains the species with lowest chromosome numbers, might be the progenitor of many of these other types. It's interesting that this genus is confined almost entirely to tropical Africa. So maybe we might say that the Vitaceae apparently originate in a tropical forest environment, and that the most primitive forms come out of this Scyphostemma, where the lowest chromosome number 2n equals 20 is found. Now, if we review these chromosome numbers of the various genera that we have here, we may hypothesize a little bit and come up with some interesting numbers as far as chromosomes are concerned. First of all, if you were to look for basic numbers that would explain most of these conditions here, we could assume that originally we had the chromosome number 5 and 6 and 7. These we call basic numbers, the ones that you begin with in this species evolution. With these numbers, you can explain all of the present chromosome numbers of the genera reported. Let's take our simplest case where we have a chromosome number of, for example, 20. You can see that with 4 of these basic sets, you could get that species there. You could get a chromosome number of 20. If we take a 5 and add it with the 6, we will get a number of 11, in which case then, by doubling, we can get the number 22. If we take then 6 and 7, we can get 13 and double that, and we get 26. So you see, all the possibilities are present. This is rather far-fetched, but it is not as far-fetched as you may think. For example, we, many years ago, used a similar hypothesis without knowing the chromosome numbers involved, and many of these species had not been determined 15 or 20 years ago. But, for example, we made a hybrid between Vitis, which has got a total of 36 chromosomes, or we might, 38 rather, 19 pairs, or 2n equals 38. And we cross this with Muscadinia, which is rotundifolia, the species we use, rotundifolia, which has 2n equals 40. So this would contribute 19 chromosomes in the gamete. This would contribute 20. So in the hybrid, we had the F1 hybrid, we had 39 chromosomes. 20 coming from the Muscadinia, the genus there, and 19 from the genus Vitis. When we looked at the pairing of the chromosomes in this F1 hybrid, we found out that the hybrid was extremely sterile due to the fact that many of the chromosomes were not nearly alike one another to bring about normal pairing. What we usually obtain was this condition: about 13 chromosomes paired together, and there were 13 unpaired, or singles. This should add up to 39 if we've done it correctly. This would be 13, would be 26 chromosomes, 13 paired, and this would be 26 plus 13 to give us our 39, which checks out all right. So one way this could be interpreted is that Vitis then has 13 chromosomes which are like Muscadinia, the other genus, since they pair together. So these two would pair together. This might mean then that originally Vitis was obtained from a 6+7, and that Muscadinia also had the same derivation to give the set of 13. Now, in addition to these 13 pairs, there were 13 unpaired. These could probably be explained on the basis that you had a pair of 6, or rather a set of 6, plus a set of 7 that were different. Call them A's or B's, that were different from each other, and one set of which were brought in from this one. You see, you'd bring in a set of 6 from Vitis to account for the 19, and you'd bring in a set of 7 from the Muscadinia. So this was present in Muscadinia, this was present in Vitis, but the sets were different from each species, so they did not pair together. One is A, one is B. So this is the type of information that may be very useful from the standpoint of how far you can go in, say, crossing and in breeding of new varieties, because we have determined that the Muscadinia has within it many genes for resistance to diseases and insects which are not present in the Vitis group. So it is very useful then to be able to know that these two genera can be crossed and that we can obtain genes for resistance from another genus. Now, despite this idea that there is a lot of duplication of chromosome sets, in Vitis perhaps the 6 and the 7, this apparently happened a long time ago, so that in the meantime, Vitis has gone over to a diploid type of condition where the chromosomes are all similar and pair together. In other words, all the 19 chromosomes, if we look at a Vitis species, pair together. So even though it might be, on the assumption it might be a 6 plus a 7 plus a 6 as the basic chromosome sets, these sets have apparently lost their differentiation, and all of them are now acting in a similar manner because of the fact that they're all pairing. Whereas if there were certain homologies left in these, you'd expect to get more chromosomes than you find in the normal pairing or bivalent condition. This means then that these polyploids, since they contain more than a single set, these polyploids must be of very ancient origin, so that in the meantime they have come to act or breed just as diploids. However, we would expect in some cases that the genes might be present instead of being present as single pairs. If you had pairs of chromosomes, you would have, say, for example, one pair of genes determining a character. If we had this condition then in the 3 sets, we may have 3 different loci for a single gene. So this complicates the breeding behavior. Now we want to leave from the back of the building. Today's lab class will meet in the back of the winery. There's a bus today. Are there any questions? About 2 minutes.
These groupings, 5, 6, and 7 groupings, I think suggested